So welcome to lesson 2 for Amplicons. This is a webinar style coffee hour that we decided to do based on some conversation among the instructors. And I guess the motivation for this is that there's a large variety of different approaches for analyzing amplicons, as there are for a lot of the different subtopics in BVCN. So I just wanted to have like an informal conversation about the approaches that different people take, because there's really no one size fits all. But what are people's preferred methods of how they treat data, how they analyze data, recognizing that the field, like all research, is an evolving field, right? And so new things are coming up every time, and we can try to incorporate that as we move forward. So I guess I will start with our document of questions. The first question here for instructors is, how important is it that we use ASVs over OTUs? And I guess whichever instructor wants to chime in and go first, you can turn off your mic.
Sure, I'll jump in first.
Mike.
So I'm not sure if we should share it here or in the notes, I guess. There's a paper by Callahan et al., who's also the developer of Data 2. So this, if you're of the specious type, you might consider this questionable because he's got a horse in the race, so to speak. But he lays out, I think, really clearly the values of ASVs over OTUs. And by that, just meaning single nucleotide resolution methods as in ASVs. And basically it comes down to that OTUs aren't really comparable across data sets if you're using a de novo approach. So I guess, I don't know how much we're going to cover here, but the two main ways of generating OTUs are closed reference. So you're using known sequences that you trust already and trying to fit your new data to those known sequences. And the problem with that is that it is very constrained to what you already know, and you miss a lot of novel diversity. The de novo approach of clustering OTUs recovers all the sequence variation that's there, but incorporates all of the sequencing error and mistakes that happen. And the bigger problem is that with even de novo OTUs, they're very determined by the data set itself, and you can't really compare different data sets. You have to go back and recluster everything together if you want to introduce more data. That's not really scalable at all. Amplicon sequencing variants try to infer real biological sequences by incorporating error and abundance of different things. And they stand alone at the end as a true biological unit, which works amazingly well. And so you can just combine multiple outputs from different data sets when you want to compare them. And that scalability and the inferring actual biological sequences, again, is all presented really well in that Callahan et al. Paper that we'll link to. And sometimes you want a lower level of resolution, but it's better to generate the ASVs and then cluster them into something because you're still working with the real unit at the start. This is the general consensus that I see and agree with.
Okay, great. So maybe a follow-up question to that, based on another conversation I actually had with Sarah, is would you, if you were a reviewer of a paper, would you reject a paper that used OTUs?
I would not do that. Definitely not. I think this is one of those things where you can be led astray with OTUs, but not necessarily. And I would never like fight somebody on wanting to use that method, but that's just me. I think some people would.
And I think the important thing that Mike's emphasizing here, right, is that empirically, one option isn't right or wrong. It's how you use those options, right? And so if you were doing an OTU study and tried to like just grab OTUs from other papers or other people's work and just compare the OTUs directly, that would be wrong, right? Like you can't do that. It's very clear that that's something that you can't do. But if you were using OTUs just to get a general broad sense of what the diversity looked like in your sample type, right? And it's one sample and you're just looking at that one particular sample and publishing on that or communicating that information, that there isn't anything incorrect about approaching the analysis from that point of view.
If I can add on top of that, the whole field of amplicons has been moving into higher and higher resolution in the last few years. So 5 years ago, you could publish a paper with 97% ID OTUs, then it went to 99%, now we're at ASVs. But the question is what you do with that data later, because if you resolve your OTUs at 97%, which is used to be thought of as species level, but then you talk about differences at the phylum level or the order level, it doesn't even matter how you clustered your OTUs, right?
Yeah, I think that's a really good point, Ella. I'm definitely one of those converts as well. I, during grad school, did the whole OTU approach a lot. And ever since I started using Data 2, have kind of converted over to the ASV style of things. But I will say that I'm actually still actively trying to publish a few papers that have OTUs in them as well. So I think we're on the fence with that. And I think probably the whole field is moving more and more towards ASVs. At least that's the feel I get from people. But I don't think we're anywhere near close to chucking OTUs out the door, and especially in terms of what you're saying, that if you're talking about things on a much higher or lower resolution in terms of phylum, then it doesn't really matter that much.
Yeah, I think that it's just important to make sure you always know what your ecological question is, and remember that I define myself as an ecologist first, not as someone who's going to like compare all OTUs and ASV, iterations. I think that like to kind of go off of what Mike was talking about and the question that Liz and I were talking about another time was, would you reject a paper for some reason just because they use OTUs? But I always ask myself, like through each part of my analysis, okay, remember that when you're looking at Amplicon data, it's like you're going in the woods with a flashlight to survey all the birds, right? And it's dark, it's nighttime. Okay, there's like inherently things that are going to be wrong with that sampling approach. Like I'm by myself, I have a single flashlight, it's at night, I'm going to miss things. I'm also going to have capture a certain like species of bird at a higher frequency than I would during the day. And so as long as the person's approach is acknowledging that and considering that and the way they write about it, I think that alone I just make sure is happening in any of these papers.
Okay, great. So I just want to mention that I am paying attention to the chat box, and so I'm trying to incorporate the questions as we go and fit them into the flow of this. So I think maybe the next logical question based on the chat box would be, since we're sort of talking about like how the field is evolving, if you still have data from a 454 data set, is that still acceptable to publish, I guess?
Well, I think this is one of those elements. I mean, so it's, again, if it's 454 by itself and you're going to approach it as with the best methods that are out there for 454 sequence analysis, then sure. I think one of the things that really stands out, and this is going to sound like we're all hyping ASVs to the highest possible level, is that there is, and recently, it's not the most, it's recent, but like not so recent if you hadn't seen it in the last couple of years. The Data 2 folks put out a particular pipeline for dealing with 454 sequences, right? So that would be one of those situations where if you had 454 and you had Illumina and you could call ASVs accurately using their recommended workflow, that would be a situation where you could actually get into a data set that then you could combine both of those outputs. Where conversely, if you tried to take 454 data and Illumina data and then make those into OTUs together, that would generally be frowned upon, right? So, the platform that you're using in this situation, as long as you're using tools that are directly addressing the limitations of that platform, doesn't limit you at this point. And so I think, that point of view of it's 454, it's old, it therefore can't be good, doesn't actually have to apply.
Yeah, I'd echo. Don't let anyone like technology shame the data you're working with. All this changes really quickly and it's still really valuable. There's absolutely nothing, there's nothing wrong with using 454 data or any data you have lying around to try to figure out everything you can from it.
Can I ask a kind of related question? Well, I don't want to interrupt someone though if they're going to comment on that directly first. So like I've been moving towards metagenomics and metatranscriptomics. I think a lot of people are, but still a lot of the first pass at analyses is amplicon. And I've almost been like more excited with ASVs and a lot of the newer statistical tools to deal with ASVs downstream. So do you see the future, like if you agree with that, like future move towards metagenomics and metatranscriptomics as like the primary sequencing approach, do you think that amplicon sequencing will continue to be as popular, or do you as like a tool to supplement that approach, or do you think it'll start to like fade or go out of fashion or something?
I'll comment first if no one else is ready to go. I think, of course, it's really hard to predict where things are going, but I've not been a fan of people being like, just do metagenomics, it's better than amplicon. I don't buy that at all. If you're dealing with, especially if you're dealing with a large number of samples, you can still reveal underlying differences between your groups and then decide what you should focus on more, just cost and high throughput wise. Right now, it still makes sense to use amplicon in lots of situations. I think it's still the first tool in the microbial ecologist toolkit for good reason.
If I could chime in too really quick, just based on Mike's point and the fact that, yeah, I think it's, if you have the money and the time, it's almost best if you can do both. If you're trying to do metagenomics to marry it with amplicon sequencing, and because you can get two kind of different sets of data there, right? Metagenomics is about kind of functional potential there. But one of the big problems with that is depending on the depth of your sequencing, you may not even recover any of the ribosomal genes there to say that, well, we've created this MAG or this bin of genes, but calling taxonomy on that is a little bit different than calling taxonomy on the amplicons themselves. So if you can do both, but I don't think you should just supplement metagenomics for amplicon sequencing in some cases for sure.
I mean, I think it definitely is all about the question that you're asking, right? And so Mike has a really good point. There's very few labs and few funding bodies and few people with the money to say, sure, go and sequence 10,000 metagenomes to the depth of coverage that you need to do this accurately because metagenomics is better. But there are ways to approach that question with amplicons in a feasible economic standpoint, right? You can do 10,000 samples, right? And so it's probably, and you know, and Mike even said this, right? It's still a really good, it's a cost-effective way of approaching your new samples, things that you're doing for the first time to get a snapshot view of what you're looking at and then make future decisions. But at the same time, like those types of kind of exploratory, small scale amplicons really only serve that purpose at this point in time, right? Like if you want to do a study that's going to blow people away that uses amplicon sequencing, you need to start thinking about an experiment or time series that has a large scale to it and that has a lot of purpose and meaning to it, right? You won't be able to necessarily do an ad hoc, you know, amplicon experiment and have it be something that's mind-blowing, but you can design a, you know, if you're in the human microbiome, you can design a, you know, multi-year sampling per day, hundreds of people type study that would have a really huge impact. And you can take, you know, same with like, you know, from an ocean perspective, right? You can, you know, you might not be able to do every sample that you ever collected from a Niskin bottle right, on a very long cruise, but you could do that with amplicon sequencing if you had a reason to, right? And so I think when you start to look at it from that perspective, it's all about the scale that you're trying to approach, and if you have an experiment that's going to give you something meaningful by that type of analysis.
I'm gonna jump in with a sort of a different, but not really different opinion. I definitely did my PhD in a lab that has been using amplicons for many years and published many great papers with that. Now I'm in a place where everybody's like metagenomics, and 16S is useless. And they do have a point to a limit, I will say, because you can, 16S doesn't always represent your true diversity. True, as if anything is actually true, right? But doesn't represent the same diversity that you would see with metagenomics, and the phylogenetics is not always the same. And that being said, I disagree with the fact that 16S is useless and that we only have to do metagenomics. I think that that's a very privileged point of view. So sure, if you can afford to sequence metagenomes from 1,000 samples and you actually have the computational capacity and the people who can deal with that, awesome, that's great for you. There are maybe like 5 labs in the whole world who can do that. So, you know, this good old tool is not going anywhere anytime soon, in my opinion.
If I could piggyback on what Ella said, I really like things like what we're doing right now, right? Because I think that we are, and a lot of other people are doing this too, bringing this science to the broader public, and like with tools like Cyverse and whatever, you can start to expand those type of analyses in places that typically would not have been able to do it when we were doing our PhDs. Okay, I'm going to move on to the next question, which is sort of related. So if you have an amplicon data set and you have singletons, do you delete them? Or do you delete them only for certain types of analyses, like diversity calculations versus like ordinations? I know Mike has an opinion on this.
I do have very strong opinions on this. The first amplicon data set I worked on, I got like 60,000 OTUs using mother 3% OTU clustering back in the day. And about 40,000 of the 60,000 were singletons. And I was like, this is probably not good or not right. And I didn't know, I don't remember what I ended up doing with that one. I think I found a Bakulich paper that said like 0.005% minimum abundance thresholds are good. And I used that and I moved on, and I was happy to not think about it anymore. But Robert Edgar, the developer of Muscle and uparse and unoise, likes to present this example where he takes the mock community of 22 organisms or whatever it is, the standard one that a lot of these tools use, and runs OTU clustering and can get tens of thousands of OTUs out of it, even at like, I think 97% clustering. And it's because there are a ton of sequences and the sequencer is introducing variation here and there constantly all over the place. And if you're getting thousands of OTUs, lots of them singletons, out of something you know has 22 unique organisms in there, I think that's actually the total number of unique 16S sequences. That just makes it very clear to me. No, OTU methods do not give you true singletons. Maybe some of them are, but a large number are not. And they insanely inflate alpha diversity metrics. If you're using an ASV approach, those often automatically integrate some sort of minimum abundance filtering because that's how they decide what they trust is true and not. And so at the end of that, you can trust sort of what you're getting. But in general, with typical OTU approaches, I think it's insane to argue that all of those singletons are legitimate and not an artifact.
Yeah, and I'll jump on top of that and say, you can have 40,000 out of 60,000 singletons that are singletons, but if you sum up their abundance, they end up making up 0.5% of your community. Then you can happily chuck them. They're not going to affect your calculations or your conclusions later on. So down with the singletons.
Yeah, I agree with both of you. I am, as a quick example of when I'm not throwing out singletons though, is I'm kind of thinking about a project where I'm going to be comparing uh, microscopy based data and OTUs and ASVs. And I think I want to see the composition of those singletons in that context of comparing the 3 different approaches. That's one thing I'm interested in seeing. I don't know if that'll go anywhere, but I'm not going to throw them out right away. I want to kind of see what the composition of that is. If anyone has any guesses, let me know.
Thank you. And did anybody else want to say anything? Okay. So maybe before moving on to my next question, let's jump into a question from the chat box. This is from Haley. I work on experiments with extremely low biomass, and we often just extract enough DNA for anything but amplicon. Are there low biomass advances? I think maybe you mean advantages for metagenomics or advances?
I can try and quickly speak to this because a lot of the stuff I've worked with, I guess, could be considered extreme environments. Most of it having to do with freshwater systems on glaciers. And for a lot of my grad research, I dealt with extremely low biomass stuff. And people can correct me if I'm wrong, but I remember a few years back, the lowest input you needed for a metagenome was something like a microgram or something like that. And that may be years and years ago, but I know now you can work with a nanogram of DNA to get a metagenome out. And maybe you're working with even lower amounts. And there are definitely ways around that, but there have been significant advances in the library prep kits for metagenomics that you can typically use to get metagenomes out of low biomass samples. And we can talk about those kind of more offline if we need to later.
Okay, great. I will put a note to get some like references or lists of kits or something from you.
Can I
Yes.
say one thing? Okay, so I used to work in ocean environments, which is definitely not low biomass, but the cells are very small and streamlined, so they don't have a lot of DNA. Maybe that applies. And then we always like to say that there's an average of like 2 femtograms of DNA per cell. So a nanogram still gets you about 500,000 cells. You can think about it that way. That being said, it's probably the place to say that low biomass environments are really, really, both for amplicons and for metagenomics, really, really prone for contamination issues when you're sequencing. So just be very aware of that if you're just starting to work on low biomass environments. That the kits add contamination, all the reagents that you use are going to have contamination, even PEG. So always sequence your blanks.
Okay, thank you, Ella. So let's move on to the next question. So now getting into like different opinions on approaches or preferences. So how do you run your pipeline? Do you use an institutional computer, a virtual machine, or do you do things locally or something else?
I usually work on a institutional computer or server, but particularly with Amplicon data sets, I've also run them on my computer locally. Things like Data2 are extremely efficient now compared to how it used to be. They don't scale, they can scale linearly as opposed to OTU clustering traditionally would not. So a lot of the Amplicon data sets I think can be done on a typical modern laptop for sure.
The real limitation there comes into the size of the data sets, right? So if you can store it on your laptop, it might be able to run on your laptop. If you can't store it on your laptop, it's probably not going to run on your laptop. Like in a reasonable amount of space. But yeah, I almost exclusively do all of my work on an institutional server. So, but there are a lot of other options out there for folks who don't have access to that type of resource. Right, we've, as instructors here, we've discussed a couple of them. But there are, I don't want to speak I don't want to speak right now because I'm not entirely sure if all of them have the ability to do work with amplicons, but things to look at are things like Cyverse and KBase. KBase might not actually do it, but public resources that allow you to use your data or other people's data and then process it are available out there for folks.
I will also chime in on this question because I recently moved from like a research university doing a postdoc to a small liberal arts college. And so we no longer had a server to work on or anything. So I switched to using Amazon Web Services. And with some very small amount of grant money, you can pay like a couple $100 a month. If you're using it intensively, I think I only went up to like $300 or $400 a month to pay for that service. And then when I'm not using it, I mean, you barely pay anything. So that's also another option.
I also want to point out, and I don't know if this is still a thing, but Amazon Web Services used to actually give educational grants to grad students up to, I don't know, $500 or something along those lines. And I think I used one in grad school as well. Maybe it's still a thing. I haven't checked in a few years. So that could also be something to look into.
Okay. I will ask another question. So in lesson 1, I kind of gave a brief overview of like the different pipelines that you can use or how you can sort of patch together like Chime with data or Data2 with some other downstream. So maybe if each one of you could go through your general like workflow, like where do you start your pre-processing and where do you end up doing your like ecological statistics?
I can start maybe. I'm also running it on an institutional server, but I'm usually starting with like fast QC and multi QC and Trimmatic outside of one of these pipelines. And then I'm running everything in Chime 2. And I'm using Data2 to generate ASVs. And then pretty much when there's an ASV table, I bring that locally on my computer and then I run everything else in R. And I wanted to add that I actually have been running all of this on with a workflow manager. So I'm using SnakeMake. So I just provide a single directory with all my raw FASTQ files, and then my end product is a ASV table. That's all the answers, yeah.
Anybody else?
Go ahead, Mike.
I was gonna say something similar. I might use CutAdapt to remove primers, or maybe BBDuke, which is part of the BB map suite of tools. And then now if I'm working on an Amplicon data set, it'll be Data2, which I run in R. And then I'll do my processing in R2. And just like Sarah said, usually when things are processed, I'll bring it to my local computer to do analysis and things in RStudio interactively.
Can I ask you a follow-up question about cutadapt?
Yeah.
When are you using cutadapt outside of R? Is that like a case, does it depend on like the sequencing quality or something? Or
I do that to remove the primers where I
but
want to, oh, go ahead.
doesn't Data 2 do that? Or
Data 2 could do it, but it doesn't find them. It does it based on their location. And often primers won't start at the exact first base like you expect them to. It depends on the
Yeah.
sequencing facility returns the data to you. And so I just find it easier to run it with something that actually looks for them rather than just cuts off the first X amount of bases.
Yeah, I think that's why I remained in QIIME 2 to run Data 2 instead of moving to R, because it was like made more sense to me to use the cutadapt in QIIME 2 and then go into the next step. But
Got you.
Um,
Yeah.
yeah, I'll go real quick. I have a very similar workflow to Mike, so you can tell I've been on Happy Belly Bioinformatics a lot too. But yeah, I was a total QIIME convert back when QIIME 1 was around. That's kind of how I cut my teeth on everything. And then QIIME 2 came out and I got scared for some reason. So I decided to go the Data 2 route and do a very similar thing as Mike, using cutadapt. And then everything in Data 2 through R. And sometimes if I'm lazy, I will just process the downstream stuff on the institutional computer, but often you move it to your personal computer, and then this is the type of thing you work on at like midnight on a Saturday night at home or something. But you know, that kind of thing. But yeah, similar stuff.
Ella?
Yeah, my contribution is a little archaic at this point. But the way I used to do this was Stream-O-Matic, merge my reads with usearch, and then throw everything into Mother. Ooh. Please tell me people are still using Mother. And the reason I did that was that at the time I was using QIIME 1, and I managed to have a problem with every single one of the clustering algorithms that were offered there. They were giving very divergent results. So, and Mother was working beautifully, so I just stuck with that. But I guess I'm gonna have to switch to Data 2 like everybody else at this point.
sometimes it's good to start with what your neighbor is using, so you can learn the pipeline.
I haven't done a full like Amplicon study in a long time, but when you grabbed some fairly small data sets recently that were Amplicon based. And so in those instances, my goal was always more or less to detect and to quantify the organisms that were there, what their recounts were and what their relative abundance was. So in that instance, I was using Data 2 combined with the most up-to-date Silva databases for taxonomy, which I think is something that's important. And you'll see a lot of, this is a complete tangent, but one of the things that is like a huge pet peeve of mine are papers that come out in 2020 that are still using versions of Silva or these 16S databases that are either old or out of date, right, by half a decade. If you go back to some of those older databases, there are entire phyla of organisms that aren't included. And so you get these like really weird assignments back. So using the most up-to-date available database is really important for that situation. And then because of what my target have been, I actually haven't gone down the route of moving the data over and then doing my own ecology analyses. So I'm not somebody who can comment on that.
Okay. since this is maybe relevant right now, this was a side question that came up. Is there a difference between calling data2 in chime2 versus calling it on its own, I guess, in R?
I can comment on that. It depends. So if you think about it, for something to become a chime2 plugin, it's kind of like making a GUI, a graphical user interface. It's phase 2 of the development of something. And so the core data2 that Ben Callahan and others develop will always be the most up to date. And then it needs to be sort of moved over to chime2. And there's almost always going to be more functionality in the core version of a tool than in some extrapolation of it or some extension of it. And I can't remember the exact example, but there were functionality that somebody needed for their data set at some point, and they were trying to process it in in chime2 using data2, and it didn't have the option that they needed, but the core data2 did. And I'm sure by now chime2 has implemented that particular option, but that sort of thing can happen from time to time. That said, if you're comfortable in chime2 and you're not comfortable in R, there's no reason you should be like, I have to learn R now to process this data set. Like, work where you're good, and if you run into an issue, then I say deal with it.
Okay, that was a great answer. Any other comments on that? Okay. Next question is so following the pipeline question, what is your favorite interface? So if you're using R, I guess many of you are using RStudio or using Jupyter Notebooks, or some of you doing some analysis in Python of Amplicon data.
I'll go first. For me, it's almost exclusively R, I guess. I think I feel most comfortable there. I'm okay on command line stuff, but my Python is really weak, and I don't really know anything about Jupyter Notebooks. So definitely some of the things that I'm going to be learning through BVCN, which is great. But in terms of Amplicon analysis, I do everything in R.
Same.
I will mention that I want to move on to doing this. I want to, because when you're like starting a project, you kind of have like this exploratory phase where you're trying all these things. And then you have like the version of things that are finalized that are going to end up going in the manuscript. So ideally, what I think would be cool is doing the exploration phase in Jupyter Labs, because it's a very nice layout of how, like, okay, I tried this, it generated this figure, I don't like it, so I moved on to the next one. And so because it puts the figures, it embeds the figures in your coding basically. So I think that's a nice way of presenting like how you explored your data. But then the final R code, if you're using R, your R scripts could just be in RStudio. That's my ideal way.
I'm actually doing something the exact opposite. I work in both RStudio and Jupyter Notebooks, but I like that the Jupyter Notebook layout, like the way you can share that with people, is like, this is what I did for my results, and I can annotate it, and then an embedded figure of like a final figure that I used or something. But yeah, similar, but not the same.
Can I ask a totally off the topic question regarding what you were both talking about? Can you take, so I've worked very little with Jupyter Notebooks, and I do think they're amazing. I just haven't spent time getting into it more. Can you share that as like a static HTML after you're done, or does somebody have to run the notebook to sort of look it up? You can do that?
I'm not sure, but I think using Binder would be a good way of sharing it.
You can definitely share it as a static HTML.
Cool.
That is a definite yes.
There's also an easy, wait, you can share it as an HTML, but can you also share it as like a R markdown and then it has a similar format? I don't know this.
I just like all the things you're talking about, except I don't want to require people to need Jupyter Notebook to like run it and launch it to look at it and stuff.
Right. No, but the HTML will give you a static version. And so instead of having in that instance, right? So if you've, fleshed it all out and had run all of the cells in your notebook, you can then export that HTML, and that preserves all of the images that you've generated and all that stuff. As opposed to if you shared the notebook, somebody would then have to run this individual cells. Yeah, no, you get a nice HTML to share with folks.
Okay. So next question is, can you share how you manage your data? Everything from your raw sequencing files, the FASTQ files, up until your OTU or ASV tables and your code and your figures. Anybody?
That is a great
File.
question. Ella, do you have any thoughts on these? I mean, I guess it's specifically of the question is, or maybe Liz, you can just clarify, is what are we doing with the data at various steps, right? Like in the final form, right? So I think, right, so like the flippant default answer for maybe raw sequences is like, oh yeah, well, raw sequences, you take them and you share them and you put them in SRA, right? And that's where they're preserved going forward. The trimmed quality control, version of those reads, I'm not sure. I mean, SRA doesn't necessarily want duplicates, right, of both, right? So it's harder to share that. If you are adamant about making sure that everybody has exactly the data that you worked with, you can store them in other kind of public repositories that are formatless, right? Something like Figshare or Dryad, right? But those have size limitations. I think the important step there more has to do with the having the raw available and then having the steps that were taken accurately to process them from raw to quality controlled, right? And then assuming that somebody else would potentially have the ability to replicate that step. And then as for getting to preserving your ASV output and your data tables, I'm not sure I have a good answer to that, but maybe somebody who's done that recently can talk about how they were preserving it. I mean, one of the worst ways to maybe do those things would be to include them as like supplemental PDFs in your final presentation, right? Like something that makes it very hard to actually capture that information. But if you're not doing that, if you're not coming up with the worst possible solution to share your data, there are a lot of other opportunities to do so. And maybe somebody who's done that more recently can say what they've done with their ASV tables and even their ASV files, right? So like if somebody, we talked about it all the way at the beginning, how ASVs are something that you can directly compare, but having access to those ASVs are pretty important to be able to do that in comparison. And so just what everybody else has been doing would be, I don't have an answer, but maybe somebody else does.
I try to put everything in like a single GitHub repo. So then there's hopefully the raw table, like the ASV table, and any metadata tables that need to be imported can live in there. And then there's a single R script. And now I'm going to have an HTML file as well that will that can be looked at and exported easily. But then if anything's too large, I've been putting it in Zenodo right now. That's like more of the reproducibility part.
Yep, similar for me too. Whatever the journal will allow you to include, I do, and then put everything else that's important somewhere that's archived forever. Like I used Figshare for a long time, and I'm starting to be swayed over to OSF, the Open Science Framework, which seems to be more friendly for having a wiki right next to it and being able to like write down a markdown format explanation of things, which I like a lot. And for Ben's example of data in a PDF, some journals will require you to do that, and it's infuriating. And just do what they say and then put the right stuff somewhere else.
I think I've included like a raw ASV table though as a supplementary table, not as a PDF, but just as another form. But I also have it somewhere online.
And I think that's important, right? Like, is that some journals are going to require you to have the data in their version of publicly available, and so it's fine if you can load up like an Excel spreadsheet, right? And that gets actually preserved in there, right? There are some journals that if you give them an Excel spreadsheet, the output is an Excel spreadsheet, and there are some that if you give them an Excel spreadsheet, the output is a PDF, right? And so even if you've done the minimum of what the journal has required, I think that step of putting the data in an accessible place, even if it is quote unquote accessible in a PDF, is a really important step.
I definitely overall would rather have journals require something like that though. If I'll definitely give it to them in whatever format they ask. It'd just be great if all of them asked.
Yeah. And then Sarah has this really good point in chat of like a lot of these options that we're talking about in terms of data storage locations that aren't something like SRA, like Open Science Framework, Figshare, Data Dryad, Zenodo. It's all about assigning a DOI, digital object identifier. Those things become almost 100% permanent on the internet, and they allow people to get back to a very specific time and place in your research and analysis. And it's okay to iterate on that information, right? And provide a version 2 that gets its own DOI and a version 3 that gets its own DOI. But you want folks to be able to access directly that point in time that you are trying to preserve for them, right? And so one of the things that I definitely will ding people on is if their data is on their own personal website without a DOI, if their data is shared through a Dropbox link or a Google Drive link. Oh, well, we created a Google Drive and that's there now. Google Drive is not required to preserve anything, right? At any point in time, you personally can delete it. Google can delete it, right? There's a lot of reasons why that things might happen. But when you put it in something that has a DOI, that's preserved for an extended period of time, if not forever. I mean, heat death of the universe, not there anymore. Collapse of electricity and the DNS backbone of the internet, not there anymore. But up until those kind of points in time, we probably, it will continue to be something that people can reference in the future.
Okay, we are at 50 minutes. So there were some questions in the chat from Slack, and I think one of them at least is like an interesting broad question that we can discuss for a few minutes, and then maybe we'll wrap it up and keep the more specific things to incorporate into other lessons. But so this question was asked also by Haley. There's this issue of compositional versus count data. And I guess the way you pre-treat your count tables matters a lot for your downstream multivariate analysis. So what ways do you treat your count tables in order to account for the fact that high throughput sequencing data is essentially relative abundance and not absolute abundance?
That's like the biggest question ever, right? And it's highly dependent on the environment that you're working in. You need something to normalize to, right? And it depends on what kind of metric you actually trust. So if you're working in aquatic systems, that's relatively easy because you can just count cells with fluorescent microscopy or a flow cytometer or however you want to do that. Or you can pretty confidently normalize to the amount of DNA that you extracted. I mean, no, that's actually not. So extraction yield really varies by the gods of extraction yield. And it could be like that the same sample would have a 90% yield in the summer and a 10% yield in the winter because the humidity in the lab has changed and the temperature in the lab has changed. And also there's, you know, just general black magic to the whole thing. But there are a lot of environments where you can't count cells to normalize. So I'm working in soil right now, and there is not any reliable metric. So what you just have to do is rely on statistical methods to draw, I guess, significant conclusions from just count tables, because there's nothing to normalize to. And this is where tools like DE-seq2 are really useful. Yeah, that's my contribution.
Yeah, I think that I've been a fan of doing some of the centered log ratio normalizations ahead of time for certain analyses, usually like cluster analysis. And I'll also test, like, I'll make the scree plot to make sure that it's appropriate on 2 or 3 axes and look at the stress for whatever I'm doing ahead of time. And there's like certain things that I think are important to do that are diagnostic to make sure that the normalization is appropriate. But at the end of the day, you know, if I think maybe other people can comment on it, but I'm still making bar plots that are like showing relative abundances, and I'll subset them to look at like a single taxonomic group because I want to, like, these are really important plots for me to understand what's going, get a baseline information about what's going on in the environment. And if I end up using those for a presentation or a publication downstream, then I just make it clear that as I'm talking about it, I'm talking about a relative abundance that I observed from my calculation. I'm not saying that this is exactly what the species is doing, and I'm not trying to make too strong of a conclusion based on that. And so I think it's just making sure that, and on the same vein, this paper, and there's some other ones that I can recommend that have other recommendations for normalization ahead of time.
Yeah, we can start collating references for that, because I think there's a couple of different like transformations that you can do before ordinations that are supposed to account for the fact that in sequencing data, you know, your zeros essentially like overwhelm the signal in multivariate analyses. But there's some pretty really straightforward normalizations like log transform that are supposed to take care of that. And so I've actually never used this CODA pipeline that Haley's mentioning, but I guess that's another approach to this. But we can go through it, talk about it, figure things out. Any other comments on that? Sorry.
I would just say I listen to the statisticians like Amy Willis and Susan Holmes and the people that care about helping biologists do things better. And we're always just talking about metrics that are outputs from inputs and decisions. And if you keep that in mind, like somebody said before, the real diversity, quote unquote, or something, we're always trying to pull out information and we're doing it imperfectly. And if you talk about your things, and these are recovered 16S gene copy numbers, not this is the richness of species in a system. I think you convey that continually, and you're like admitting, yeah, this is the output from arbitrary decisions in lots of places, and I'm trying to listen to the people who know math better to do it right. That's really the best we could do.
Okay, I think we should stop there because it's been almost an hour. So hopefully we answered a couple of questions, or maybe we made more questions for some people. But that will conclude lesson 2. So thank you everybody for participating. And I hope that we can maybe build some future lessons based on the issues that came up today and follow up based on this. Okay, thank you.
That's great, thanks.
Thank you.
Nice little BVCN fireside chat.